My name is Martyn Dade. I'm a reader in design computation at Newcastle University. My background was in architecture and then in computing and interaction design and data visualization. And more recently, I became interested in synthetic biology to the extent that I persuaded my university to give me a sabbatical for a year and to pursue an MSc in synthetic biology. And so my more recent work is in particularly in bacterial synthetic biology, so looking at these organisms from the molecular level right up to the level of materials. And my aim is to build bacteria-based materials and architectural systems. And you have seen from Simon's talk that you can treat bacteria as individuals, but you can also treat them as, in effect, multicellular organisms, and that gives us a huge range of possibility. They can also do these extraordinary feats of material engineering, and so my interest is in that. Now, I don't have time to go through all our projects today, but I wanted to just give you a sense of the overall pattern of the things that we're involved in. So the project that is gaining me most notoriety and is in fact where we have most of our funding at the moment is one called computational colloids, or we nickname it thinking soils, which is to find a pressure-sensitive gene within bacteria that can be used to trigger a process of material synthesis. So if you imagine that you're constructing a building on a soil which has our bacteria in, it would make materials in response to those mechanical forces. We group our other projects into, so we go from sensing with our computational colloids project to actuation, which I'm going to talk about more in a moment. We're interested in morphogenesis, or in bacteria terms, pattern making, and how bacteria make patterns and the forms that they make and how we can influence and control those forms. And then ultimately material synthesis, and we're particularly interested in calcium carbonate production and using bacteria to make biocements, and actually having control over the way in which those biocements are deposited, and also the sorts of crystals that they make as well. And in fact, that's an area that my wife is working on now in her lab. She's a molecular biologist. Underpinning those are some general questions that I wanted to share with you, which I think are pertinent to this group. One is of scale, and it's an obvious one when you're dealing with microbiology, and particularly in synthetic biology, where we're looking at things that start at the scale of 1 nanometer, so the scale of individual molecules, genes, and then go up to the scale of a cell, which might be only 2 micrometers across, and then the architectural scale that we're used to working at, the body, the building, and the city. And for me, there's a gap between those scales, so we lack often the representations and the design methods to allow us to bridge from one set of scales to another. So while in our molecular biology lab we have a huge amount of tools to deal with that end, and in my studio we have a huge range of tools and representations to deal with this end, we often don't have a good way of bridging the gap between the two, or interfacing those two ranges of scales. We're also dealing in a world in which, in design terms, we like to look for formal elegance, and we like to design systems that are controllable and understandable, but we're using materials and systems that are radically complex. So in synthetic biology there is this push towards effectively mechanizing biological systems, understanding them as parts of systems that can be decomposed and recomposed. But the reality of taking a bacteria from one environment into another, for example, is that they behave incredibly differently, their genes respond in different ways. So we have on the one hand an idea of decomposable structures that can be made into parts, rearranged, and produce new things and can be standardized, to a situation where we have structures that are not decomposable, where everything is in some way related to everything else. And that radical complexity is something that is a real design challenge, but it's also a design opportunity. My also grand aim, perhaps arrogant aim, is to sort of build a new kind of building science. And I invoke the idea of Sir Christopher Wren, who was both an architect but also in the UK founded our most venerable scientific institution, the Royal Society. And that idea that an architect could be an astronomer, could be a physicist, could think in those multiple ways, is something that perhaps it's hard to imagine now, but I have the aspiration that we might be involved not only in applied research but also in fundamental science as well, and that we have something to offer fundamental science. And the other theme is of biomaterialism. So the work of my group, we always say that we don't do something that we can't make, so we try to make wherever possible, and that comes with its own challenges, because making in a lab-like environment is very different from working in a studio or a workshop-type environment. But we do this regularly now, so my studios are partly conducted in micromolecular biology labs. This is the hands of one of my dissertation students who spent six weeks this year working directly with microbiology and making materials. And through that making, we gain a kind of understanding, a tacit and often intuitive understanding that gives us insight into those things which are more formal research publications and so on. Just for the group in the last 5 minutes, I wanted to share with you one particular project which I think is a good bridging project, and one that we've started in the last year but has already shown some real potential. In fact, I delivered a paper on this a few days ago. Using bacteria spores as a way of creating hygromorphic materials. Now, for those of you who know about this kind of world in architecture, have come across this idea before, but hygromorphic materials are materials that will change their shape in response to water in their environment. So they may change their shape in response to humidity, for example. In architecture, there is some very well-established work now using wood-based hygromorphs, where you take a passive layer, a type of wood that won't expand when it's exposed to water, with an active layer, wood that will expand. You put those two layers together, and as you get differential expansion, it pushes one out of one layer pushes the other, and you get a change in form. And doing that, you can create buildings which have flaps, for example, will open or close when it rains, and you can get materials that are responsive also to changes in humidity. And we're working with a bacteria-based version of this material that actually uses bacteria spores. Sorry, the reference seems to have gone on my slide. That has first been developed at Columbia, and some work has been done at Hiroshi Ishii's lab at MIT to develop this material. It uses the bacteria spores, which are the simple way of saying it, and Simon will probably disagree with my definition here, but it's a seed-like form for the bacteria. It's what the bacteria does what certain types of bacteria do when the environment is not conducive for their survival. But it's also incredibly hygromorphic. But it's only about 1 micrometer across, and we use a particular strain of this bacteria that has it's modified so it has its outer coating removed, so it expands by about 17% of its size when it's exposed to high humidities. And by placing it on a passive layer, in this case a polymer, we can make a material that changes its form. And so by patterning the bacteria spore solution onto the surface of the material, we can create different forms and interactions. And then we can add those forms and interactions together to create effectively muscles that will contract as they dry and then begin to relax as they wet. And we do experiments with these things on a very basic level. So we make up the spores, we arrange them on the surface of the material. And we get our students to explore them and experience them and work out how they work and take some basic measurements, and then to do design work with them. But they're very, very challenging to work with, and they challenge us a bit. They challenge oops, for some reason that one then disappeared. So what you should be able to see there is an actuator. It was there before. It's an actuator that was designed. It's not a video, no, it's just an image, but some of the images just seem to disappear on this projector for some reason. Never mind, I'll go to this one. I can show you pictures of lots of this work, by the way, if you want to see it afterwards. I'm sure there'll be conversations that are had. But one of the challenges that often our students think in industrial terms about this, so they think about this as being an actuator, as you would get a mechanical actuator. But the forces that we're dealing with are actually very, very small, so they are very sensitive to very slight changes. The actuators themselves each behave slightly differently because of the way that we've made them. So the idea that you can just get a bunch of these things and move something mechanical is a lot more challenging. And what we've realised is that we need to really rethink this sort of material, come up with new form factors, and work actually at a different scale to start with, thinking about different sorts of structure. Just to conclude, I just wanted to point out the names of both my research group, which includes microbiologist, civil engineer and architecture postdocs and doctoral students, my collaborators in science and engineering, and also the studio, because a lot of the work is initiated by our students. Thank you.
Thank you.